Aloha again, everyone. This is Tony Gerlach. I'm excited to be with my next guest here today. A couple years ago, the Society actually invested a lot of money into research, and I'm lucky today to have our two researcher winners from the Weill Research Institute.
Hi, my name is Scott Weiss. I'm a pediatric intensivist from the Children's Hospital of Philadelphia.
Sheila Alexander. I am an associate professor at the University of Pittsburgh School of Nursing and Critical Care Medicine.
Well, great. Thank you guys for being here. Can both of you summarize your research and with the grant and how you think it's going to impact the critical care community?
Sure. My research focuses on pediatric sepsis. Sepsis is a dysregulated immune response to infection that results in organ dysfunction and too often death. There's exciting new evidence that suggests that the intestinal bacteria, or the bacteria that live in our intestine called the intestinal microbiome, can help to regulate our body's immune system, and that alterations in the intestinal microbiome can change the way we fight infections. So one potential mechanism that explains the connection between the intestinal microbiome and the immune response is the secretion of short-chain fatty acids from the intestine into the bloodstream. And these microbial-derived byproducts could then affect the way our own bodies produce energy within mitochondria. So to date, I've really focused on the role of mitochondrial dysfunction in pediatric sepsis, but through the Weill Research Grant, I'm looking to expand this work to look at the possible connection between the intestinal microbiome and immunometabolism in pediatric sepsis. And our hope is that by expanding the paradigm of critical illness in pediatric sepsis to include the non-human intestinal microbiome that lives inside of all of us, we can start to identify novel therapies.
No, that's quite interesting, and it's also quite interesting as you go out as how the microbiota affects even regular illnesses that might not be critical care illness, such as immune diseases such as Crohn's disease and stuff. And I think it's such a complex thing. We are only at the tip of the iceberg with our knowledge of it.
Absolutely. I think our model focuses on sepsis, but I think some of the things that we hope to find will be more broadly applicable.
Awesome. And what about you, Sheila?
So I look at omics of brain recovery from acute illness and injury. Specifically, this project will look at DNA methylation of inflammatory genes and changes to that methylation over time in patients who do and don't develop delirium. So I think this audience knows delirium pretty well, and it's associated with a lot of negative outcomes. There's a lot of individual variability. We don't have great ways to treat because we just don't know what's happening inside the body very well with delirium. So this work should help us understand some of the genetic and epigenetic factors that might be driving that pathology that we see expressed as delirium signs and symptoms.
Awesome, and that sounds quite interesting too. Now with delirium, you tend to think a little bit of elderly people more at risk for it. So are you limiting it to any sort of age population, or is it going to be a quite broad sample size?
So we are looking at a couple different ICUs. There's a medical ICU and a surgical trauma ICU, and I think the strength of that is we've included subjects 18 and older, so we are able to look at both the young patients that we see in the trauma ICU, but also the older patients in the medical ICU, and hopefully we will capture a good number of those so we can at least get started laying the groundwork to figure out what changes, and there may be age-specific changes in methylation driving delirium.
No, and definitely it sounds like there's differences in the population, but that's a good thing to start out broad. Now, can you guys tell me about the application process? How easy was it, or how hard, or what are some lessons learned we could tell members of our audience?
So I actually found the process fairly easy. I should note that I have served on the scientific review committee in the past, not this year or for the last couple of years. Certainly since I submitted. Before that, I have not sat on that committee, but so I was kind of familiar with the process, but it's pretty clearly laid out on the website. There is a specific link. It gives you directions, and there is actually some guidelines to help you write a better grant and things that you need to make sure you address. So I thought it was fairly easy. It's an automated system, so you can upload things, and it was fairly intuitive. I thought it wasn't hard at all.
And
Yeah,
you, Scott?
I would echo those sentiments. I think the revamped SCCM website really does a nice job of laying out the mission of all the different grant mechanisms that they offer and provide step-by-step instructions to guide you through the application process. In my own sort of application, I really started early and sought a lot of guidance from my collaborators and my mentors to try to hone my proposal to make it more clear and focused before I ultimately submitted. And I would certainly encourage folks who are interested in pursuing some of these grants to certainly start early and review the different grant mechanisms and try to find the one that fits best. I think what's was most exciting for me is that most of the time when you apply for grants, you're competing with a broad range of different disciplines, but SCCM really provides an opportunity to submit critical care research ideas to critical care-minded reviewers. And I found that to be especially exciting.